We will be hearing from Dr. Elizabeth-Sharon Fung and Dr. Jennifer Macke from the Los Alamos National Laboratory, and they'll be presenting the LANL HIV databases. So handing it over to you, Elizabeth-Sharon. Thank you.
Thank you. Thanks, Nina. And thank you, Nina, for just keeping us all on track with a smile. My name is Elizabeth-Sharon Fung, and I'm here to present with my colleague, Jennifer Macke from the Los Alamos National Laboratory. We have an HIV immunology database that is run out of here, and we would like to share some of the work that we do and things that we are able to share with not just HIV people, but also other infectious disease experts, and really with anyone who researches any organization, any organism. What I'd like to start out with is thanking IEDB. Thank you so much. We really, truly appreciate your inviting us here annually. And we do like the opportunity to share the work we do and how we can help the rest of you. So thanks for that. Our database PI is Brian Foley, and we have a whole team that supports us over here, and we are funded by NIH. One of the things I'd like to start with is basically, how does one get to our database? So what you have to do is go into your browser and type in hiv.lanl.gov. That's all it takes, and you get to our database front page. You can see here that there are two pillars on which our database rests. One is the sequence database, and other is the immunology database. And this is, of course, an HIV database. Our HIV database over here has information in both places, but the information can be got to from either side quite seamlessly. It does not, it isn't hard at all.
Elizabeth-Sharon, I think that your slides are not progressing.
Oh, okay. Thank you. Let me see what's going on here. Do you see it going on? Is it progressing now?
No, I think you need to choose a different view.
So like we see the back end of your presentation, not your presented slides.
Is that,
yes.
Is that better? Okay,
yes.
So I'll dial back just a few seconds. This was our title page you probably saw. And here's the next page. This is how you get to our database, hiv.lanl.gov. Here's the sequence tab and the sequence button and the immunology button, and that's where you can get into information in our database. So what is it that our database covers? What do we have to offer you? In short, we have data to offer you that has been curated, but we also have tools that you can use to go ahead and mulch this data, to massage it, to look at it in new ways, analyze it, present it in different ways. And these data and tools, if you can be looked at in the context of host pathogen interactions, which I know many of you are infectious disease experts, so this is interesting. How can we find both these bits of information in the database? One, under the pathogen side, you can find things like HIV sequence. We have links to HIV structures in PDB. There is modification of that sequence that we have information on. And we also have linear or discontinuous antibody epitopes. On the host side, we also have CTL or helper epitopes in the context of restricting HLA, their immune response. There's a lot of information on antibodies, as well as interactions that they have with HIV, or also neutralization that it has against HIV. And then we have a patient database which actually connects to much of this information. Experiments may be done with certain patient cells, or also certain HIV subtypes and viruses or variants have been found in different patients. This is a kind of information that you can find in our database. Also, all this information or the tools that we have are presented to you. But one important thing is that the tools are not just HIV specific for the most part. Here's a list of a few of our tools, and the red buttons are the ones that actually are HIV specific. So you can see that most of our tools can actually be used for other organisms or for other for your own research that you can put in, and you can use these tools. So If you're not an HIV person, please do visit our website, because there is a lot we have to offer you as well. The other thing that I wanted to talk about was the things that we do in order to keep all this information current for you. We have continuing efforts, but we also have some new upgrades for this year. And that some of those, just to touch on a few, are that our patient database is now searchable and has been expanded. The HLA nomenclature is now consistent with the 2022 update that came out. There is an N-glycoside tool that has been updated. We now put in information on somatic hypermutation of certain antibodies, and this can be found in our neutralization database. This is our neutralization database, which has a lot of data as well as some tools, is called CatNap. So there's synonyms here on our database. And then the API for JSON downloads have been updated and also expanded to cover variants. So HIV variant data can also be downloaded in this fashion. And if we were to go back to our database, I want to show you where you would find some of these bits of information and links to them. So I'm just going to show you the immunology side. So if you were to click here, this is the page that would come down, and you can see that you have a main menu that is a drop-down menu at the top here. You can go to the different databases, sort of sub-databases. You can perform searches, look at tools and products. You can do the same thing here. Click to searches. There are lots of products, which include maps, tables, things like that, and then also tools and a few other things. But today I would like to take you very rapidly through a few epitope searches and show you how these can be used sequentially. I'd like to start with a linear antibody epitope search and end with a discontinuous one, but show you how you can go from a search to using a tool. And in this case, I'm going to talk about the HIV Genome Browser tool, and then just do a few overlapping searches of other epitopes. So let's get started with that. Back to the front page of our database, if you were to click on immunology, and then you would go to this page that I showed you earlier, I'm going to choose antibody search here. One could find the same kind of information by going to epitope or antibody tables, but let's go here. And this is an antibody search page. What you would do here, I want to show a linear antibody epitope search, and the antibody in question that I've chosen is a common, well-known one, 10E8. It's a human anti-HIV antibody. So if you were to type that in here in monoclonal antibody name, and then hit search, the next slide shows you the results. The results are a page with notes, and then I'd like to point your attention here to the epitope. You can see here that there's a logo that shows you the relative usage of this epitope with different amino acids in HIV across its variants. There is also over here the location of this particular epitope within HIV sequence, and that becomes very important. It's one way of defining the epitope. I do want to point your attention to the fact that all of our epitopes and much other data is standardized against one consensus HIV sequence, which is called HXV2. And this really helps to standardize the information across the field, because you can go in and find where your epitope fits in against this well-used standard consensus sequence, and the extension number of that is given here. But I'd like to take you to see where this particular antibody epitope lies on HIV sequence. And to do that, we're going to go into clicking to an epitope map. In this case, it's GP160, which is part of ON. And you can see it's a very long, big map. There's a lot of information on it. We're going to focus in on this region because that is where our epitope of interest lies. And then I'm going to zoom in and show you what that looks like. So here's just our little panel that we're interested in. And you can see already that 10E8's antibody epitope lies in this region of HIV. So you can follow the lines down and find what that particular epitope is and where it lies on HIV, in this case, HXV2. But you can also see that there is another well-known human antibody 4E10, which seems to its epitope overlaps 10E8. So that's interesting. So maybe we need to go take a look at it some more, and I'm going to do that using a tool, the HIV Genome Browser. So if you were to scroll to the top of the page and look at it, right at the top, there's a link to Genome Browser. And so what we've done is we've gone from a linear antibody epitope search to looking at a map, and now we're going to a tool to look at more things. So click on that and you get to this page. You would then need to select ONV because that's the protein within which this epitope lies. And then you get to this page and on the tools. And over here on the left, there's a menu from which you can select what track you want to look at against the HIV protein sequence up here, which is numbered according to HXP2. And so I'm going to say I just want to look at antibody epitopes. So double click there and I'm going to zoom in. And then you have, this is what you see on the genome browser tool. And because I've already looked at it and made a slide, these open arrows here denote where exactly the 10E8 antibody epitope is. When you hover over it, you get this little box that tells you that it's a 10E8 epitope. This is a sequence. If you were to double click, you could get more information. But instead, what I would like now to do is to look at the sequence and see, is there any other information that we have about neutralizing antibodies, which 10E8 is one, and context around it? Is there something more I can find out about regions around or overlapping this epitope through this tool? So I double click here, and the next slide, we will see the neutralizing antibody contexts as well. And that is seen over here. So you see these pink antibody epitopes. I've zoomed in a little bit. That's 10E8's epitope. And here these indigo boxes show you different contact positions within different epitopes. And what I've chosen to show are these clear arrows, and the information from them is here in the boxes. But I'll just show you one of them, this first one, which is from this arrow here. So that's right to the, this portion of the 10E8 epitope. And if you were to look at that, you'd see that what it tells us is that if you were to mutate positions 672 and 673 to a leucine, then that neutralization of this 10E8 antibody would be affected against HIV. So that's the kind of information you can find here just by looking at context. But I'm now going to minimize this, take this track off just so we can see other things. So we're back to just the antibody epitopes. And this time, I'm interested in looking at the HXP2 sequence and seeing, are there any sites of interest that have not to do with neutralization, but other sites of interest on HXP2 that overlap with my 10E8 antibody epitope that I'm looking for. And so click on that. And the next thing you see is, well, there are some nearby. And these are these purple lines over here, which show you different sites of interest. You can see that upstream of the 10E8 antibody epitope, there is a heptad repeat. Interestingly enough, there is nothing to see that is overlapping with the 10E8 antibody. However, if you look just downstream of it, there is a transmembrane domain, a GP41 transmembrane domain, which is very interesting to someone that studies antibodies, to say that 10E8 binds just next to a transmembrane domain. This is an interesting thing for people to know. So this is the kind of information you can find here. But let's now minimize that track and go on to seeing other things. So back again to the antibody epitopes, but this time I want to look to see, are there any overlapping helper epitopes or CTL epitopes that overlap the same position on HIV along with my 10E8 epitope? So click both these, and you click both these, select them, and on the next slide, this is what you would see. So our T-cell epitopes are here in green, the helper epitopes down here, the CTL epitopes here, and just for reference, some of them overlap the 10E8 epitope as well. So let's say that we want to look at one of the helper epitopes. What I've chosen to show you here is this one, and you can see here with the little pop-up box that comes when you mouse over that it does overlap the 10E8 epitope, and it does not have a defined HLA restriction. If you wanted more detail, you could double click on that and look at it here. So now let's look at a CTL epitope that overlaps. And here I've picked this one here, and you can see here that this one does have a restriction that has been published, an HLA restriction, and there's more information here. Suppose I were to want to find out more information on this, I could zoom in and I could copy this particular sequence, which is the CTL epitope that overlaps the 10E8 antibody epitope, and then I could go on and do a CTL search. The CTL search is done in a form that's very much like the antibody search that we had done and started out with, but it's a different form, of course. And so that's right there on that front page. So put it in the epitope and click search, and the result is notes again. These notes have a lot, but what I'd like to point to your attention is that we do have a logo, and then the positions are given. And in this case, we already know that the restricting HLA is A0201, but we also have patient data for this. So there is a patient who had a response to this and this particular epitope, and they have been fully typed. So if they have been typed for HLA, then the patient's complete HLA typing is given over here as well. However, before I move on to the next thing, I wanted to show you that there is also an observed variant or a variant place over here. This means that there is published somewhere a variant to this epitope. And if you were to click to this, you could find more information, notes, and just places on maps for those variants. So I will not go into that, but that is available as well. So let's go on now to the last antibody set of searches. These are discontinuous epitope searches, so not one linear epitope. And the one I chose was a mouse anti-HIV. And this guy, if you were to put his name in and then click search, the results are I've given right here. And you can see that it is discontinuous. There are two separate portions of epitope that are over here, and that you can see there's two of them. Now let's go on to seeing a different one. And this one I've chosen human PG9, which is a very well-known antibody, anti-HIV antibody, and search that. And this time on the results, you can see that there are actually different bits of information you can find. One that I'd like to point out to is that there are neutralization details available. This is in the neutralization/CatNap database. And Jenny Macke, my colleague, will be talking much more about that. So I'm not going to talk about that, but know that you can go from these antibody notes to the neutralization data that is associated with this antibody. And then also here, there are contacts and features. That is another database we have that is associated with the antibodies, where it shows you what different points the antibody contacts HIV. So this is very important for discontinuous epitope antibody. And that result looks like, this is from the features database, something like this: a table where there are different amino acids that are shown to have contacts and different features on the HIV sequence. But I'm not going to go into that in detail. Jenny will have a little more to say about that. All I wanted to say is I've shown you now how you can go from an epitope search to pathogen attributes using things like protein maps, a tool, and looking at things within the tool like sites of interest. We've also looked at how you can go from an epitope search to host attributes like antibody stuff, where you have antibody contacts, you have antibody neutralization, and putting these two together, you can have a lot to say about host pathogen interaction, which ultimately helps to inform vaccine design and even other things, even drug design and things like that. And with that, I'd like to end and say thank you for letting us present. And we always have a need for ideas for tool development. Please hit us with them. We love that. And this is how you can actually contact us if you have questions of any kind. Please do so. And I will stop sharing now so Jenny can continue. Jenny Macke is my colleague, and she knows a lot about the antibody side and the sequence side. So take it away, Jenny. Bye-bye.
Oh, thank you. Good morning. Let me just get my screen share going. So I'm going to give you a quick introduction to some of the resources that are available from the sequence side of our database, with a particular emphasis on resources that would be of interest not just to HIV, but beyond HIV research, and also those things that may be of particular interest in immunology research. So this is just the beginning, the entry page to our sequence database. If you have used it, I'm sure you've seen it. Our main menu contains links to pretty much everything that would be of interest. We'll take a quick look at the database tools page. Similar to the immunology tools page that Elizabeth-Sharon just showed, the tools are color-coded to denote which ones are HIV specific in red. If you're not working with HIV, certainly you'd be looking for the tools that are marked in blue or purple, and that is the majority of our tools that are usable for other organisms. The first resource I wanted to go into a little bit is something Elizabeth-Sharon touched on, which is our database of antibody contacts and features.
Excuse me,
those,
Jenny.
yes.
I think yours are not progressing either.
Oh.
So it's the slideshow tab on the top, I think.
Okay, is this showing the tools page?
It is.
Okay, but I need to go.
It's showing the back end of your presentation, not the presenter.
Okay. And does this look right? This should be showing the tools page on a reader view. No?
We're still seeing the non-presenter view.
I'm not sure why the other view wasn't working. So yes, this is the tools page, as I mentioned, with the color coding. And then Elizabeth-Sharon mentioned that we store information about discontinuous contact residues and binding sites in a separate database, which you can access through searching for a specific antibody, but you can also go to this database directly and download, if you want to, the entire database of contact information and other features with an emphasis on neutralizing antibodies. So the other features would include things like sites where a mutation affects neutralization, for example. Almost all of the data in this database is based on published data, not on predictions. There are a few features that are predicted features, but those are clearly marked by the experimental method or by the title of the feature. This data is used by a number of our other tools. It is pulled into our antibody searches, it's pulled into CatNap data, and also into the genome browser, as Elizabeth-Sharon just showed. And this just shows the output page for looking for one particular feature. This is the contact sites for the well-known antibody VRC01. And I hope you can see my cursor, but where it says envelope position, that is the HXB2 coordinate position of the feature. And
Jenny,
yes,
it's not progressing now.
I don't understand.
Is it possible that you have multiple windows opens with this, and that we are maybe if you
It's a funny little thing.
so that if you have another window which is advancing. So right now we're seeing slide 4, and we don't see your cursor moving or anything. So
Okay.
I suspect you're sharing it a separate instance of your application. Now we're seeing your cursor, and we're seeing view neutralizing antibody contacts and features.
Okay, okay, good, I think.
You were going to try going forward once and back to see.
Okay,
It does advance. This looks good.
okay, I apologize for the problems. So yes, this is the output page showing the contact sites for VRC01, the envelope position of each of those contact sites, the other features, what's going on at those contact sites, and the entropy at each of those sites. Entropy meaning variability of the sequence. All right, so moving on, one other tool I was going to give a quick intro to is our Analyze-Align tool, which is for analyzing variable sequences. It has some built-in features for HIV research. So if you're looking at HIV, you can use sort of preloaded or pre-available alignments. But if you're working with another organism, you can provide your own alignment. And in this example, I've taken an alignment of HCV E2 protein and put that into the input. You can choose which region of that protein you want to look at in detail. So I've selected residues 11 to 22. The selection that you choose could be the location of your favorite epitope or an epitope-rich region, or just a region you want to look at in more detail for its variability. And then the other option I've selected is I've chosen to use the first sequence of my alignment, which is a reference sequence for HCV. And then I hit submit, and hopefully this advanced to the results. So the first, it gives the output in 4 different forms. First of all, a sequence logo showing how variable each of those positions is. The second is a list of the variants present relative to the master sequence. Next, a histogram of how many changes each of those sequences had. So 74% of those sequences had no change relative to the master sequence. A smaller percentage had 1 change, 2, etc. And finally, it gives an alignment of each of those variants at those positions relative to the master and their frequency. So again, 74% of them had no changes. And then it shows the next most common variant in that alignment, etc. So this is just a quick look at one of our tools and how it can be used for organisms other than HIV. Another set of tools that we wanted to touch on are our vaccine design tools. We know from HIV research that epitopes can be very densely packed. So it's very difficult to, say, optimize one particular epitope in a vaccine design. So the general philosophy or approach of these tools is to consider every possible 9mer to be an epitope, and to try to optimize in the vaccine cocktail the coverage of all of those naturally occurring 9mers. So the philosophy is that it's not necessary to know the epitopes or to predict them, but just to deliver them in an intact way, and to prioritize the fidelity of all possible epitopes of the protein. So we have 2 tools designed to accomplish this end. The first was the Mosaic Vaccine Designer, which was published in 2007. And the second is Epigraph, which has the same goal, but uses a completely different algorithm to approach the problem. In both cases, we're trying to generate candidate vaccine cocktails that optimize the coverage of possible T-cell epitopes, not predicted epitopes, but just taking every part of the sequence as a possible epitope. The suites also include evaluation tools to show you how well a prospective vaccine cocktail will cover those epitopes. So first, this is the input page for the Mosaic tool. The input that it requires is an alignment of naturally occurring protein sequences of a diverse population. The things that you can choose are the cocktail size, how many protein sequences to generate, and you can play with the the epitope length that it's trying to keep intact, with 9 being the default. The Epigraph tool is very similar in terms of input. It requires the same input and has some of the same selections available, but it uses a very different algorithm, which should be fractionally better. And it's also much faster, so it allows more iterations of the data in coming to its set of optimal proteins in the cocktail. I don't have time to go into really a lot of detail on those tools, but just to mention they've been used in many studies, and some of the studies of HIV that have used these tools have gone on to human clinical trials. And you can see here a list of all the other organisms which have also used this approach to design vaccines. And the other tool I wanted to touch on is CatNap. compile, analyze, and tally neutralizing antibody panels. So what is that really? It's basically two things. It's a database of neutralization data, IC50 and IC80 data taken from publicly available sources. And it's a tool to analyze those data or to analyze other numerical data in association with sequences. And just to mention, we are working on a SARS-CoV-2 version of this tool, which we hope to have public in the next month or two. So the tool page provides several different options or different ways to approach the tool. The standard form, the first one there has all that built-in IC50 and 80 data and allows you to analyze it. If you wanted to combine that data with your own anti-HIV neutralization data, you would use the hybrid version, which is the third one on the list. And the version that I'm going to show an example of is the custom input. So on the custom input, you can input any numerical data in association with sequences and do a similar type of analysis. I'm going to show the output page from the tool, and I know this, if you haven't looked at a catnap analysis before, this may be a little bit busy and overwhelming, so I'll go through it just to show you the highlights. So the data I've put in this case is IC50 neutralization data for 5 neutralizing antibodies of SARS-CoV-2. The names of the antibodies are right here in the center across the top, 5-7, et cetera. And the actual neutralization values are shown below each antibody. And on the left are the names of the viruses that neutralization data is for. And then on the far right, the sequences of each of those viruses. And if this were a live window, I could scroll from left to right and look at the entire length of the protein sequences. Below those windows, the tool calculates the geometric mean IC50 of the detected viruses or of the detected and undetected viruses and the percent of detection. So antibody 5-7, for example, has neutralized 29% of these viruses based on this data, whereas this antibody on the far right was able to neutralize 100% of them. The color coding indicates the strength of the neutralization, so red and orange is more potent neutralization. Below that, we have a position analysis that we can do. So when I put this data in, I noticed that these antibodies neutralized these first 2 viruses. And coincidentally, those first 2 viruses have a glycosylated asparagine at position 17. The tool will highlight in red asparagines with predicted glycosylation. So I ran a quick position analysis here on position 17 for antibody 5-7. And in the second box, it shows a statistical prediction of how likely that is to be by chance. Again, I should emphasize that this doesn't tell you with any certainty that this glycosylated residue is associated with that particular antibody. You would need experimental data to really know that for certain, but it can show areas that are worth exploring and potential linkages between what an antibody neutralizes and what's going on in the protein sequence. The final thing I wanted to mention is that some of our colleagues are working on this COVID-19 genome analysis pipeline, which has many of the same tools as the HIV database site, but customized for analysis of SARS-CoV-2 sequences. The sequences in this website are updated almost daily. So if you are working on keeping track of variants as they come in real time, this has the most current data that are available. There are a lot of different tools that you can run, and I can't possibly show you all of them, but just one really quick example. This is the Analyze a line tool that we looked at just a moment ago from the HIV site. Here on the SARS site, it gives some specific options for research on CoV-2. For example, you can choose a specific lineage to narrow your search to, or you can look at just one particular geographic region of interest and look at just the variance and the variability of the sequence for a specific lineage or specific geographic region. So that's the end of what I have to present to you. Thank you very much for listening to us, and please get in contact with us anytime with questions. We do answer the questions that come on our help email lines. So thanks so much for having us, Nina.
Excellent. Thank you so much, Jennifer. Thank you, Elizabeth-Sharon as well. And apologies for a few technical issues. It doesn't matter how many times we practice and try, Zoom always tricks us. So I hope that was, you still managed to get all the information you needed from that. So thank you. And now we do still have about 8 minutes or so left for the Q&A. And we also have the break afterwards. So if we need a little bit of extra time, that's not a problem. So now I would like to invite Jay back, Elizabeth-Sharon and Jennifer. I think there are a couple of questions and I would normally say let's start with Jay, but there are no questions on the API just now. So Elizabeth-Sharon or Jennifer, did you want to take the first one?
Sure, I can take the first one, which I've already answered, but I will say it out loud here. And the question was, what does ONV stand for in slide 4? And that is HIV envelope, which is the main, well, it's the outside protein of HIV. And so it's the one to which most antibodies are generated. And then, of course, there are many CTL and helper epitopes also off of ONV. So that is ONV. And Jenny, I think the next one, I don't know if you've had it.
I will read the question. I think I would need the person who asked it to clarify a little bit more exactly what they mean. It says, I like the features because I have modeled a vaccine target. I'm curious to how it might resemble fair certainty via the antibody. And I think I'm having trouble understanding exactly what the question is, if it's about the feature database or about vaccine design. So I would encourage, this is a question from Mark McGarry, I'd encourage you to write your question to our Seek Info email helpline, and we can have more of a back and forth discussion of what you'd like to know.
If you'd both like to just drop that email written as a typed answer into the chat after not the chat, the question, then Mark can definitely reach out. Thank you.
Okay, we can do that. I can take the next one, which is the Mosaic tool only for CTL epitopes? And no, it also generates helper epitopes, but though it is formally for T-cell epitopes, some of the scientists here have actually run it for antibody epitopes. And if it's a good structural region, they've actually been able to find antibody epitopes too. So formally for only T-cell epitopes, but has been found to have a good response for antibody epitopes as well. Jenny,
yeah, the next one is the entropy data also available for other viruses, i.e. SARS-CoV-2, in a specific genome region? That's a great question. It might be available directly on the SARS-CoV-2 pipeline website, but if that's not something that they are currently providing, we have a tool called entropy, where you can take an alignment of a variable organism and get out the same kind of entropy data that we were showing for for HIV. So go to the HIV site and look for the entropy tool, and you can get that kind of data out in a nice format.